group mentored by Dr. Dave Pittman.
Hello all and welcome to our senior thesis presentation. We are Dr. Pittman's group. I'm Gabby.
I'm Morgan.
I'm Vivian.
I'm Katie. And I'm Kennedy.
And our study is mixed drinks or straight alcohol. Adolescent rats don't care. Both increase consumption. So we used previous research to model human alcohol consumption. Our first model was our Bay group, our brief access to ethanol exposure, and this modeled our binge drinking behavior, or the acute intake of alcohol over a short period of time. And our second group that we looked at was a chronic intermittent access group, which is the low to moderate intake of alcohol over a longer period of time. Previous research has shown that the brief access group does increase the intake of alcohol over increasing ethanol concentrations. But previous research used the chronic intermittent access group, which gave rats about 4 to 6 hours to lick the alcohol. And we wanted to give them a little bit more time, so we did our continuous access to ethanol group, which exposed the rats to 24-hour free access to ethanol over a longer period of days throughout the week. Previous research also used adult rats. In our study, we used adolescent rats because the binge drinking behavior is first seen in adolescents with human teenagers. We asked 2 questions in our study. Does brief and or continuous access to ethanol in adolescents increase the consumption of ethanol? And our second question is, does pairing a non-nutritive flavor with alcohol increase the consumption of that non-nutritive flavor?
So in our study, we used 28 adolescent male rats, which we split into 3 groups. The naive group, which received no access to ethanol. The Bay group, which received brief access to ethanol, and the K group, which received continuous access to ethanol. In the Bay group, we put them in the MS-160, as you can see in this video, and the shutter will open and the rat will lick a variety of 6 different concentrations of ethanol with orange flavoring or water. Of those 6 different concentrations, 3 of them were less than a percentage of a beer, and 3 of them equaled the percentage of wine or a mixed drink, all the way up to 40%, which would equal a shot of vodka. The K group was given a 2-bottle consumption test in which they were given water with orange flavoring or 10% ethanol with orange flavoring equaling a mixed drink. As you'll see in our results, it looked like the rats might not like the orange flavoring, so we decided to replicate the study without the orange flavoring. In phase 2, the naive group was put in the MS-160 and given the same procedure as the Bay group in phase 1, without orange flavoring. And then the Bay group and K group from phase 1 were given a 2-bottle consumption test without orange flavoring to make sure it was not confounded or aversive.
Now, looking at the results, on the y-axis over there we have the lick ratio, which is a ratio comparing the number of times the rats licked ethanol to the number of times the rat licked water. And on the x-axis we have the increasing concentrations of ethanol. This graph compares the lick ratios of the Bay 1, Bay 2, and K groups between the pre-test and the post-test. So as you can see on the lower end of the graph, we had a shift up in acceptance for all 3 groups between the pre-test and the post-test, and that was specifically at the lower concentrations. So this means that with exposure time, the rats became more accepting of the lower concentrations of ethanol. You can also see there was a similar trend in acceptance for all 3 groups, regardless of the type of access to ethanol they received. There was no significant difference in acceptance between the Bay groups and then the K group, so this suggests that the type of exposure to ethanol does not significantly affect the acceptance of it. However, we did find that at 40% on the pre-test over here, that the Bay 2 group licked significantly more than the Bay 1 and K group. But then on the post-test, the K group licked significantly more than Bay 1 and Bay 2. And between the pre-test and the post-test, there's a significant increase licks for the K group, but not for the 2 Bay groups. So this means that only the continuous access to ethanol ended up increasing the acceptance of the highest concentration of ethanol. Again, we have here on the y-axis the lick ratio and the different solutions along the x-axis. This graph shows the EC50 values for rat BZ23 across the first 8 days of exposure to ethanol. The EC50 value is the concentration where the orange solutions and the ethanol solutions produced half of their maximal response to water. And you can find that point where the lines cross at 50% along this dotted red line. The 1 on the y-axis is how much water the rat licked, and then the numbers below the 1 mean the rat licked less ethanol than it did water. So that shows that the ethanol was more aversive to the rat. Now, along the dotted red line, you can see that for each day, the EC50 value shifted to the right, and this shows that this rat became more accepting of the higher concentrations of ethanol as exposure time increased. You can see on day 1 that the rat was accepting of around 1% ethanol, but by day 8 it was at around 15%. So you can see that significant shift over time. Ultimately, calculating the EC50 values allowed us to quantify acceptance over time and make some insightful comparisons in our experiment.
Here we have the EC50 percentages for the bay groups of phase 1 and phase 2 over a 12-day span. The orange dotted line is the group that had the orange flavored ethanol, and the black dotted line is the group that had plain ethanol. On day 1 over here, that's not working that well, but we can see that there is a significant difference between the two groups. This could be due to an immediate aversive reaction to the orange flavoring, but we really can't be sure. We did have a main effective days and a days by group interaction. After day 1, the rats are trending more towards that 10% concentration. Then over the course of the 12 days, they're trending more towards that 15% concentration. In the middle here, you'll see this little gap of like a 3-day cluster where there is a significant difference between the 3 groups, but we didn't find this to be meaningful. We just think it is due to variability of the groups. So overall, a takeaway from this graph is that there was not a significant difference between the 2 bay groups, which just suggests that the orange flavoring was not like a confound as we initially thought. And here we have the 2 bottle preference scores for the K group in phase 1, the bay group in phase 2, and the K group in phase 2. That red dotted line across, if the lines were trending around that red dotted line, it would show that the rats were consuming equal amounts of ethanol and water. But because these lines are trending below the red dotted line, our rats were consuming around 30% ethanol and 70% water. The takeaway from this graph is that there was no significant difference between the 3 groups, so that just suggests that the prior access the rats had to ethanol, whether that was brief access or continuous access, did not significantly impact their preference for ethanol in phase 2.
So what does all of this mean? When we first began our study in February, the purpose of this study was to examine whether brief access exposure to alcohol or continuous access exposure to alcohol in adolescent rats would affect their alcohol use behaviors when they became adults. Throughout our study, we were able to find 2 key takeaways that we think are important to share with y'all. Our first key takeaway is that even though the rat did not like the orange behavior, or the orange flavor was not a confound and did not negatively affect any of our data collection. In fact, as a thesis group, we have suggested to Dr. Pittman and his next thesis group to use other flavors of non-nutritive flavoring instead of orange flavoring to see if the rats prefer other flavors instead of orange. Our second key takeaway was that we were not able to support the previous model of binge drinking found in rats. This previous model states that brief access to alcohol would increase their binge-like drinking behaviors as an adult. However, we found through our study that there were no differences in alcohol use behaviors between rats that were in the brief access exposure group and rats in the continuous access exposure group. Instead, we found that these rats all treated alcohol the same way when they became adults from their adolescence age, and we suggest that this could relate to human behaviors as well, that overall exposure to alcohol as an adolescent leads to an increase in alcohol use behaviors as an adult. We would like to end our presentation with thanking our rats and Dr. Pittman for all of their contributions to our project. Thank you. Does anyone have any questions?
Let's hear the back. The reason you all selected male adolescent rats only?
There's no specific reason. We just did not, in order to have a significant analysis, we wanted to have enough rats, and if we did male and female rats, we would need 28 of both, which we were unable to use in our current laboratory equipment. So we decided to do 28 male rats. However, Dr. Pittman has posed in his next group to possibly use female rats instead.
Did you talk at all about how the alcohol consumption might change the brain and like basically like what the mechanism would be to either increase or keep the same level of alcohol consumption whenever they go up.
As a thesis group, we didn't discuss this. However, one of my research papers for my research paper talks about the addiction reward system in the brain and how, as an adolescent, when you're first given that exposure to alcohol, it changes. And so you become, it's similar to substance use in humans. The rats' reward system changes and they become more addicted to alcohol and thus increase their alcohol use over the increased exposure to alcohol.
Yeah, I definitely think that would be interesting. We didn't look at neurobiology, we also didn't look at behavior, even though we did notice some things, but nothing as drastic as we thought we would see, but there were some really interesting studies.